Hello.
Thank you very much. Can you hear me well?
Yes.
Okay. Can you also see my mouse?
Yes.
Okay, good. So I'm very sorry that I cannot be in Krakow in person due to my health issue, but I'm very glad that I can participate at least virtually. So I'm going to talk about robust and reliable compartmental models of neurons. And you know now already how to construct such models from the talks by Dieter Jäger, Bruce Graham, Arndt Roth. So now we are going to look at their robustness and reliability. So unfortunately, many published models behave unrealistically when they are used in different morphologies. So if you switch the morphology, sometimes the models fail, or if they are used to reproduce data that they were not tuned for, so outside of their scope. And many contain a mixture of ion channels, sometimes a wild mixture of ion channels from different species. This is also one reason why they typically cannot account for species differences. And there are other problems that have been nicely summarized in this review by Almog and Korngreen in 2016, in which they asked the painful question, is realistic neuronal modeling realistic? And also several technical issues you already heard about in the talk, in the previous talks, for example, a talk by Dieter. So we know that neurons and neuronal dendrites are very diverse, as you saw also in the talk by Hermann. So the dendrites come in different shapes, sizes, diameters, and morphology changes during development, during neurogenesis, after lesion. And there is also an astonishing biophysical diversity in ion channels. So different neurons have different numbers of ion channels with different kinetics, and they can also change. So there is also intrinsic plasticity. You can observe this in knockout experiments, in pharmacological experiments. But despite of this morphological and biophysical diversity, neuronal behavior is surprisingly robust. And our models should reflect that. So our models should be also robust despite of the diversity. So maybe there is a need for a novel kind of compartmental models that would be robust to morphological and biophysical changes. We are not the only groups that develop such models, but this is the motivation why we joined our efforts. So with Hermann, we are using his trees toolbox, the morphological modeling toolbox, in combination with Neuron that you learned a lot about already. And we have a trees to neuron interface. So these tools allow us to explore morphological and biophysical diversity. I want to show you one example for a morphologically robust model. Here we developed a model of hippocampal dentate granule cells. You can see here the 3D morphology reconstruction. And you know now already that you can take these reconstructions and you can import them into Neuron and you can run simulations. And so you know that there are basically 3 steps. If you create a compartmental model, you need morphology, then you need to insert passive channels and active channels, active meaning voltage dependent, calcium dependent channels. And that's what we did here. So after an extensive literature search, we inserted 15 ion channels into the dendrites of granule cells. So on the right, you can see 3 different morphologies, examples of reconstructions, and on the left, you can see the expression of ion channels. These black lines, their thickness represents the expression of a given ion channel. So for example, a sodium channel NAV 1.6 has a high expression in the axon initial segment of granule cells. And this model was able to reproduce several electrophysiological data sets, patch clamp data sets. So I'm going to show you only some examples. So on the left, you can see experimental data. So here you can see IV curves, voltage plotted against current. And you can see that the model in reconstructing morphologies can replicate the data well, and it also can replicate a pharmacology experiment. So if you apply barium, it blocks certain ion channels, and this shifts the IV curve. And interestingly, this was possible to do also in the completely fake morphologies generated with Hermann's trees toolbox. So you now understand better how we created them using these optimal wiring principles. And even in these completely fake morphologies, the models were still able to reproduce experimental data. Here are some examples for the number of action potentials plotted against the injected current, again for the control condition and for the application of barium. And here are some examples for voltage traces. So the model was able to capture not only the number of action potentials, but also their shape, their amplitudes, their widths. And interestingly, we could convert this model from a mouse model to a rat model. So we started with a mouse model, mouse data, but then when we switched the morphologies, so at first the model was not performing very well. So you can see here this blue line, dashed line. So if you just switch the morphologies, then the passive properties are not reproduced very well anymore. However, when we adjusted one channel, the Kier channel, so we increased its expression 2.5-fold, and then we were able to reproduce red data. So this is a bit speculative. So these simulations predict that the higher expression of Kier channels might account for the species differences between rat and mouse with respect to their passive properties and also their firing. But this has to be still confirmed in the experiment. And I don't want to go into detail here. I just want to show you that by adjusting the ion channel densities, you can sometimes create a new model that is able to capture new phenomena. So here we adjusted the ion channel parameters according to the literature, and we were able to simulate not only the behavior of mature granule cells, but also of the young, newly born granule cells. So maybe you know that the dentate gyrus is the structure in which there is adult neurogenesis still going on, and these young granule cells are more excitable as compared to mature granule cells. So you can see a leftward shift in their input-output curves. So I would like to sum up this first part of my talk. So you can download this model, you can use it in your own research, you can extend it, improve it. So I showed you that we created a new granule cell model that is robust across a wide range of reconstructed but also artificial morphologies. And after adjustment of passive properties, we were able to convert the model from a mouse model to a rat model. And also, after adaptation of ion channel properties, we created a data-driven model for adult-born granule cells. So far, I ignored biophysical diversity. So the morphological diversity was there because in our simulations, we used different reconstructions, different artificial dendrites. However, in all these models that I showed you so far, we inserted the same ion channel density in the different sections or compartments of granule cells. So then in a follow-up project, we wanted to explore biophysical diversity. So that's why we created, by using random parameter search, we created a database of models. And this is an approach that you heard about in the talk by Dieter. So Dieter Jäger mentioned database modeling. It is sometimes called also population modeling because instead of a single model, you create a population of single cell models. This is a bit misleading because population models, sometimes people use it also for network models, but here we mean populations of single cell models, or sometimes people call it also ensemble modeling. And not surprisingly, we discovered ion channel degeneracy. So this is an important term. If you are not familiar with it, I can explain it to you. So ion channel degeneracy means that different configurations of ion channels of their parameters can lead to a very similar neuronal behavior, to very similar neuronal output. And you can see it here. So different colors here represent different models. So we started with our realistic granule cell model, and then we randomly changed the parameters, and we wondered what happens if we keep the biophysically full model, and what happens if we remove some channels. So we created also reduced versions for this model, a 9-channel version and 5-channel version, and also within these reduced versions, we varied stochastically the parameters. And you can see here very similar firing patterns generated by different combinations of ion channels. And this you can see in these plots below. So the y-axis represents the 0 to 2-fold change in ion channel expression with respect to the initial model. So 1 would mean the same ion channel density as in the initial model, and 2-fold change would mean 100% increase, and 0 would mean a knockout of a channel. And you can see that different colors represent one channel for one model that is a valid model. And you can see that the ion channel parameters are spread all over the y-axis. So there is a wide range of possible ion channel configurations leading to the same output, which is in line with this idea of ion channel degeneracy. And this has been discovered by Yves Marder back, I think, 20 years ago. And so interestingly, not only the biophysically fully complex model was able to reproduce spiking features, but also the reduced models were able to do that. So on the right, you can see the spiking features that we tuned the models for. So for example, firing rate, spike latency, or fast after hyperpolarization, and so on. And so also these reduced models, a 5-channel model was still able to reproduce realistic firing. However, when we generated these random models, of course, we generated not only valid models, but we generated also many models that were not valid. And when we looked at the percentages, interestingly, for the full biophysically full model, the percentage of valid models was higher. So we observed that there was almost 6% of models were still valid, as compared to the 5-channel model. There we observed only roughly 1% of valid models. So there is a relation between the number of ion channels, number of intrinsic mechanisms, and the chance of getting a valid point in the parameter space. And that was true not only in the range, in the 0 to 2-fold range, but also when we sampled the parameter space in a closer neighborhood of the valid parameter space. So for example, in the plus minus 20% range, there we observed a similar phenomenon. So you can see here that for the 15-channel and 9-channel model version, we get about 80%. So about 80% of models are valid models. And for the 5-channel version of the model, only about 30% of models are still valid. Then we also were wondering whether the biophysically full model is more stable or robust as compared to the more simplified, to the reduced models. So for that, we performed a random walk through the parameter space away from the initially valid model. And we counted the steps away from the valid model. So you can see here these asterisks denote the mean number of steps away from the valid model when the valid model became a non-valid model. And the number of steps that you can walk through the parameter space is higher for the biophysically full version of the model. So that means that the valid parameter combinations, so that the full model can endure a more higher number of perturbations, so it is more robust. And also when we knocked out channels, so we observed that the biophysically full model was able to compensate better. Maybe not so surprising, but it was interesting that when we deleted any calcium channel or any potassium channel, so for example, here you can see a spiking pattern for the biophysically full 15-channel model version. If you remove N-type calcium channel or the BK potassium channel, there were still models that were able to generate realistic firing. So that was not the case for the 5-channel model, which is not surprising because in the 5-channel model, there was only one calcium channel left and potassium channel left and some leak channels left. So I would like to sum up the second part of my talk. The biophysically full 15-channel model of granule cells can be reduced successfully to 5 ion channel model without the loss of somatic spiking. Random sampling in the parameter space leads to about 6% of valid solutions in the biophysically full model, but only about 1% of valid solutions in the reduced model. And deletions of ion channels can be better compensated in the full model, in the biophysically full model. So now I'm talking not about morphologically full models, but about biophysically full models. And so now comes the speculative part. So we would like to generalize this maybe to non-neuronal systems, because you can see evolution as maybe as a random walk through the parameter space related to optimization. And so maybe biological complexity can facilitate evolutionary tuning of the functional parameter space. So the complexity or the degeneracy that I mentioned might contribute not only to the robustness of biological systems, but also to their evolvability. Actually, there are these theoretical ideas, for example, in this article by Whitaker and Bender from 2010. Okay, now I'm coming to the third part of my talk. And so the second part was about biophysical diversity, and now I'm coming back to morphological diversity. So here we studied whether morphological variability affects neuronal firing. And we know from previous studies that this is the case, that, for example, the neuronal branching pattern can affect firing. Symmetric dendritic trees can support regular firing, whereas asymmetric dendritic trees can support bursting. We also know that the size matters. So the firing rate depends on the size of the cell. If you inject the same current into the soma of a small cell, you can depolarize it much more easily as compared to the same current injection to the larger cell. So this is well known to electrophysiologists and also modelers. You can explain it using Ohm's law. So if you inject the same current I to the cell, then it matters what total input resistance this cell has. So a small cell has a small number of leakage channels in the membrane, and therefore its input resistance is higher, and therefore the depolarization, the voltage change, is larger for the same current injection as compared to a larger cell which has a smaller input resistance. Well, you can use sometimes, instead of resistance, you can use input conductance, but it's basically the same principle. And so I want to show you an example of using compartmental modeling to address the following question. How is neuronal firing rate and firing pattern affected by the diversity in dendritic size, but not in the case of somatic current injection, but in a more natural case of synaptic activation? So here we analyzed morphologies of different shapes with the help of Hermann's trees toolbox, and here we analyzed their passive properties. So you can see here a plot. The y-axis is the input resistance, and the x-axis is the total dendritic length. And not surprisingly, larger cells have a smaller input resistance, and small cells have a large input resistance, consistent with Ohm's law. If you consider that these cells receive the same density of synapses, so this is something like a thought experiment. So if there is a same density of synapses, then of course large cells have a higher number of synapses. And so if you drive these neurons synaptically, these two factors cancel each other out. And that's what we observed in our simulations. So here you can see a neuromorpho org-wide analysis. So we downloaded reconstructions from neuromorpho, and then we we ran some passive simulations here. So the y-axis is the somatic voltage response, and the x-axis is again the total dendritic length. And you can see that the voltage response is largely independent of the total dendritic length. So I should mention that we normalize the average diameters to 1 micrometer here, and we use the same passive properties in all these neuronal trees, the same specific conductance. And Alex Bird, our mathematician, he also derived analytical solution in a semi-infinite cable. If you inject distributed current, then you can find a very simple equation to predict the voltage change. So you see it depends only on the distributed current and the specific membrane conductance and the diameter, so that's the D here, but the total dendritic length falls out of the question of the equation. So the voltage response of the cell is not dependent on dendritic length. So our numerical simulations were in line with this analytical prediction. And so synaptically driven responses are robust with respect to dendritic size and also dendritic shape, dendritic branching. So if you make these plots and the x-axis is the number of branches, you get very similar plots. And you can derive then equations not only for distributed current injections, but also for synaptic models using synaptic conductances. So now you know that there is a difference between a current injection and the synaptic models in which you use real conductances. That was also mentioned in previous talks. So you have these saturation effects, and you have phenomena like shunting inhibition, etc. However, the equation is not much more complicated. So it's basically the synaptic current divided by specific membrane conductance, and you have again the diameter here, and you have here in addition the synaptic conductance. But otherwise, again, the total dendritic length is not anymore present in that equation. So the somatic voltage response is independent of dendritic length. And that was the case not only for passive models, but also for active models. So we showed this, for example, for these Meinen and Sejnowski models that are very well known in the field. So we took these canonical models and their spiking mechanisms and morphologies, and first we reproduced their simulations. So if you inject somatic currents, then you see different numbers of action potentials, and the larger cell generates a smaller number of action potentials, and smaller cells, like here this L4 stellate cell, generates higher number of spikes. And you can see that there is also a different spiking pattern. So this L3 pyramidal cell and L5 pyramidal cell, they have a bursting behavior. And the L3 S-spiny and L4 stellate have a regular spiking behavior. So there was a difference in spiking pattern. So when we stimulated these cells using synaptic stimulation, we saw that the number of spikes was very similar. So the output was independent of the size of the dendritic tree of these cells. However, the spiking pattern remained preserved. So you can see that there is still bursting for this green cell and this violet cell, and the regular firing for the red cell and for the blue cell. However, the number of spikes is 6 for all these cells. So we call this observation a principle of dendritic constancy, and it is valid not only so it is valid for passive, responsive, and also for spiking, but only for firing rates, but not firing patterns. So then we validated these observations also using other active models to make sure that it's not specific to the Meinen and Sejnowski models. So, for example, we used the model from Tim Jarsky, and here we again activated the synapses of different strengths, and we see that the responses of these models were largely independent of the total dendritic length. And surprisingly, even when we stimulated only a subset of synapses, so not all of them, but just rather small clusters, the responses were still largely independent of total dendritic length. So that was surprising to us. And we also used a well-known model from Jota Poirazzi, which is able to generate dendritic spikes. And here again, still we observed somatic responses, somatic spikes, largely independent of total dendritic length. And here we did not study this systematically, but even the number of dendritic spikes was largely independent of the dendritic size. But we would have to study this more in detail. But it's an interesting observation. So I would like to sum up this third part of my talk. I showed you a principle of dendritic constancy, so which predicts or shows that neuronal cells are able to electrotonically adjust their neuronal excitability so that their response to synaptic activation is independent of size. And this is valid for spike numbers, but not spike times. So spike numbers are largely independent of dendritic length and branching, but not spike patterns. And we derived also simple rule of thumb equations, which can help computational neuroscientists to set their properties, the membrane conductance, if you want to achieve a certain firing rate or a voltage depolarization. This can be quite helpful in modeling. I would like to stress that we don't show that dendrites do not matter or that all cells are equal. So that would be a very simplified message. So we see dendritic constancy as a passive equalizing backbone that is present in all dendrites. And this passive backbone can be used to maintain the responses of cells independent of size, but the responses can be changed, of course, by local plasticity rules, local intrinsic plasticity changes, etc. Now, in the next part of my talk, I'm coming back to this term of ion channel degeneracy. So like I said, the ion channel degeneracy means that different configurations of ion channels, their different ion channel parameter combinations are able to perform the same function or yield the same neuronal output, similar neuronal behavior. But this leads to an interesting question. So if you create these databases of models, so you have big populations of models, however, it's not clear which ion channel parameters are present in real neurons. Probably not all of them, not all models that we generate are really present in real brains. And in this part of my talk, I would like to argue that evolutionary ideas might be helpful here, because you can imagine that evolution removes suboptimal phenotypes from the parameter space. But first, let's have a look at this picture here. So this should show you this issue of degeneracy. So on the left, we have a parameter space. Just notice that the x-axis and y-axis of this plot, of this schematic, are parameters. And on the right, you see the performance space. You can call it also functional space or phenotype space. So what x-axis is a function A or task A or objective A, and y-axis is a function B. And the degeneracy would mean that different parameter combinations can contribute to similar phenotypes in terms of the performance, right? The degeneracy should be distinguished from redundancy. So redundancy can be defined as is the phenomenon when multiple identical elements perform the same function. So not structurally different elements, but identical elements. So we should not confuse those two terms, degeneracy and redundancy. Now, you can argue, I think, plausibly that evolutionary selection can remove suboptimal points from parameter space. So on the left, you can see parameter space, which is occupied. So you can see that the data points fill the large part of the parameter space, and the evolutionary selection might simplify this parameter space. Of course, this selection should be based on some performance, and the theory would predict that the selection would be based on the performance for the trade-offs between multiple tasks, because usually neurons and neural circuits have to fulfill more than one task. So if you look at this in the performance space, on the left, you see different phenotypes, and evolutionary selection selects the phenotypes that perform best for the trade-off between the function A and function B. And such phenotypes that perform best for the trade-offs for two functions are called also Pareto optimal. So you can see it here in this picture. We are again in the performance space. So we have a task A. You can imagine this could be dendritic computation, for example, generation of dendritic spikes. And the y-axis is another function or objective, for example, energy efficiency. And all the Pareto optimal phenotypes or neurons would lie on a Pareto front. And those phenotypes cannot be outperformed for these two tasks simultaneously by other phenotypes. So the phenotypes that have a lower performance would be removed by evolution. Sorry. So for example, the neuron 4 or a circuit 4, so this is, you can apply this also to circuits, would be removed by evolution because its performance for the trade-off between dendritic computation and energy efficiency would be worse as compared to, for example, phenotype N3. So phenotype N1 is very good at dendritic computation, not so good at energy efficiency. At N2 is very good at energy efficiency, so it saves a lot of energy, but it's not so good at dendritic computation. However, all the phenotypes on this Pareto front have a very similar performance for the joint for the combination of tasks, and N5 is physically impossible. And interestingly, the dendrites. So you heard the talk by Hermann about dendrites, and he mentioned these two laws of Cajal. And it appears that dendrites might be Pareto optimal for the two objectives that Hermann uses to generate his morphological models. So he mentioned the wiring economy, or the minimization of dendritic length. So you can see it here in this figure B. That's from a review. So wiring economy at the x-axis and signal propagation on the y-axis. So the Pareto theory would predict the dendrites would lie on the Pareto front. So some of them would optimize the economical wiring. So they would look like these blue cells here. So the dendritic length would be minimized, and other cells might prefer fast signal propagation from dendrites to the soma. But the theory would predict that they would lie somewhere on the Pareto front that optimizes the performance for these two objectives. There are papers that, so there is one paper showing that this might be the case. However, it still needs to be studied in more detail. But some data hint that this could be the case for neuronal dendrites. So I want to show you next interesting thing, namely now we switch from the performance space to the parameter space. And in the parameter space, these Pareto fronts have very interesting geometrical properties. So in the parameter space, and for two tasks, so you have two parameters here and two tasks. It doesn't have to be so. You can have three tasks. So the number of parameters and the number of tasks can be different. However, in the parameter space, all Pareto optimal phenotypes, either neurons or also other biological systems, should lie on a line. So that was discovered by Uri Alon in their paper Choval et al. In 2012. And it might be interesting to apply these ideas also to neurons and neuronal circuits, which has not been done yet so much. But here you can understand better why optimal neurons should lie on the line. So if you have two tasks, so then you have two peaks in the performance. So you have two peaks here, and these performance contours show you the phenotypes with similar performance. And the further away you are from one peak, by the way, these are also called sometimes archetypes. So the further away you are, the lower your performance for one task. And then all the Pareto optimal phenotypes lie on this line. Why? So if you look at, for example, phenotype N4, its distance from the archetype 1 and archetype 2, so that's the peak performance for task 1 and task 2, or task A, task B, its distance is larger than the distance of the N3. So N3 is on the line, and the distance of N3 from N1 and from 2 is smaller than the distance of N4. And therefore, evolution, the theory predicts that the evolution would have removed N4 from the parameter space. So if neurons are optimal, then they should lie on a line if they are supposed to fulfill two tasks. If there are three tasks, then they should lie in a triangle. And you can use similar geometrical reasoning to derive that. So all Pareto optimal neurons would be inside the triangle, and the specialists that are very good for task 1, task 2, task 3 would be close to the vertices or corners of these triangles. So this has been shown by Uri Alon, so they used it to interpret non-neuronal data, and it would be interesting to have a look at this also in neuronal models. And you can generalize this for the n tasks. For n tasks, the Pareto front is a polytope with n vertices or n corners. So here is one example how this could be applied to neurons. It's a speculation based on modeling. So it's a preprint paper, but a very interesting one, in which they generated a database of models for L5 pyramidal neurons, pyramidal cells. And so this has not been studied yet, but if Pareto theory is right, then optimal neurons should lie on a line in the parameter space. And so in this modeling paper, Bast and Oberländer showed that probably layer 5 pyramidal cells are very good at dendritic computation and energy efficiency. So they really seem to care about the trade-off between energy efficiency and dendritic computation. So then the prediction would be that they should be close to the Pareto front, and that should have a shape of a line if there are these two important functions. And this seems to be the case if you look at the densities of ion channels that people measured in these cells. So these cells have a certain potassium channel density and calcium channel density in their dendrites, and they seem to optimize this in terms of the energy efficiency and dendritic computation. So, but the details are not so important. I would like to promote here the idea that Pareto optimality might help find models with optimal ion channel configurations that would perform best for a trade-off between multiple tasks. And this could be energy efficiency, which has been shown to be important in the brain. Maybe you are familiar with the ideas of Simon Laughlin, and some functional effectiveness. And sometimes we know the functions of the neuron, sometimes we don't know, but I think these ideas might be helpful. And the hope is that this would reduce the high-dimensional parameter space to geometrically more simple, low-dimensional manifolds. So I found only one paper that used these Pareto optimality ideas in compartmental neurons, at least with respect to functional effectiveness and energy efficiency. And that's this paper from the group of Susanne Schreiber. The first author is Michiel Rem, and they tuned the models for medial superior olive neurons. And interestingly, their best tuned model, which can you see here as a star, is actually located on a Pareto front, which is here plotted for the trade-off between the performance and energy efficiency. And the performance here, again, this is a detail that is not so important here, but so here performance is defined as the coincidence detection on the dendrites of these neurons, because this is important for these medial superior olive cells. By the way, I should mention that Pareto optimality and multi-objective optimization has been used previously. So there was one landmark study by Charles Druckmann in which they used these ideas to generate, to optimize neuronal models. And it's also used in the BluePy-Opt. But it would be interesting to extend this to other functions and also to include the energy efficiency. And I wanted to show you one more example for to make a link to machine learning. So this idea might help improve maybe also artificial neuronal networks, because biological neuronal networks don't have a problem with catastrophic forgetting. So maybe you are familiar with this, that if a network learns tasks sequentially, then it is a challenge to remember old tasks and still learn the new tasks. And so it seems that the neuronal networks are optimized for a trade-off between plasticity and stability. So if the plasticity in the network is very high and stability is very low, then you can learn very well new tasks. If the stability is high but the plasticity is low, then you can remember very good the old tasks. So for a continual learning, to avoid catastrophic forgetting, probably you need a balance, an optimum between plasticity and stability. So the networks that would be on the Pareto front here in this middle region might be the best networks for avoiding the catastrophic forgetting problem. And maybe this could be inspiring for artificial networks. Okay, I think I will skip that. So I would like to sum up this fourth part of my talk. And the summary is very simple. Pareto optimality based on trade-offs between multiple objectives. For example, the functionality versus energy efficiency, or effectiveness versus economy trade-off might be used as a guiding principle for disentangling neuronal diversity and degeneracy, and for improving the population or database models of neurons. So how am I doing with time? I think now I should probably finish.
Yeah. I wrote on chat. I wrote on chat.
Fine. So I think I'll skip this last part, and I would be happy to hear some questions.
All right. So do we have any questions?
There's one on Zoom. Maybe Peter, do you see the chat on Zoom?
I can click on the chat. Yes, I can. Okay, now I can see it.
Can you read the question?
Change its shape. So, a brief question regarding the random sampling of configurations. Does it make sense to test all these hypotheses using a drift in the configuration, like studying the effect of a small change in one ion channel configuration on the number of other ion channels? For me, random sampling feels like a brute force approach. Yes, so random sampling is is basically a brute force approach, and that's also what Dieter Jäger mentioned. This database modeling or population modeling is a brute force approach, but still you can learn new things from it. And so I hope that when you then apply these optimization ideas to this, if you combine it with the brute force approach, then you can understand the valid models much better.
Okay, do we have any more questions? Okay.
Yeah, I don't see any other questions in the chat.
Do you hear me?
Yes, I can hear you.
So is there any evidence that Pareto optimality could be dynamically changed in the neurons? So for example, with increased supply of energy or oxygen in the brain, would they change for more optimal computations than from energy efficiency?
Yes, that's an excellent question. So there are not too many papers studying that, but recently there was a paper from the group of Nathalie Rochefort, a neuron paper in which they used they measured electrophysiology and neuronal properties in the, I think it was visual cortex of mice, and they were food deprived, so they manipulated their energy intake. And these mice performed slightly worse for visual tasks. So there was a decrease in function, but interestingly, the firing rate of neurons was preserved. So for some reason, the neurons cared a lot about preserving the firing rate, in terms of the firing rate homeostasis. So I think that is a very interesting experimental system in which these questions might be addressed.
Okay, so thank you very much. Unfortunately, we don't have time for more questions, so thank you for the talk.
Yeah, thank you, and greetings to everyone. I hope to see you next year in some meetings.
Thank
Bye.
you.